Infectious disease genomics is a field that explores the genetic material of pathogens, like viruses, bacteria, and parasites, to better understand how they cause illness. By studying their DNA or RNA, scientists can track how diseases spread, evolve, and sometimes become resistant to treatments. This approach has revolutionized our ability to respond to outbreaks, offering insights that traditional methods simply can't provide. In this video, we'll journey through the basics of infectious disease genomics, its applications, and its impact on global health. The heart of genomics are DNA and RNA, the molecules that carry genetic instructions in all living things, including infectious agents. DNA is like a blueprint, while RNA often acts as a messenger. By decoding these molecules, scientists can identify unique features of each pathogen. This helps in distinguishing between different strains and understanding how they adapt to new environments or hosts. The study of these building blocks is foundational to all genomic research in infectious diseases. Genomics has transformed our understanding of infectious diseases. By analyzing the genetic code of pathogens, scientists can quickly identify the cause of an outbreak, track its spread, and even predict how it might change over time. This information is crucial for developing effective treatments and vaccines. Without genomics, our responses to new and emerging diseases would be much slower and less precise. Sequencing is the process of determining the exact order of DNA or RNA in a pathogen. Modern sequencing technologies can read millions of genetic letters in just hours. This rapid analysis allows scientists to compare different samples, spot mutations, and monitor how a disease is evolving. Sequencing is a cornerstone of infectious disease genomics, enabling real-time surveillance and response. Pathogens are constantly changing. Through mutations and genetic recombination, they can adapt to new hosts, evade immune systems, or become resistant to drugs. Genomics helps us track these changes in real time. By comparing genetic sequences from different times and places, scientists can map out how a pathogen is evolving and predict future trends. When an outbreak occurs, genomics can quickly reveal the source and transmission routes. By analyzing samples from patients, scientists can determine if cases are linked and how the disease is spreading. This information is vital for public health officials to contain outbreaks and prevent further transmission. COVID-19 pandemic showcased the power of genomics. Scientists sequenced the SARS-CoV-2 virus within weeks of its discovery, enabling rapid development of diagnostic tests and vaccines. Genomic surveillance continues to track new variants, helping guide public health responses worldwide. This case highlights how genomics can change the course of a global health crisis. One of the biggest challenges in infectious disease control is antimicrobial resistance. Pathogens can develop resistance to drugs, making infections harder to treat. Genomics helps identify resistance genes and track their spread. This knowledge is crucial for developing new treatments and managing existing ones more effectively. Genomics accelerates vaccine development by revealing the genetic makeup of pathogens. Scientists can identify targets for vaccines more quickly and design them to be more effective. This approach has been key in responding to emerging diseases, reducing the time from discovery to deployment. By comparing genetic sequences from different locations, scientists can map how diseases move through populations. This helps identify hotspots, track international spread, and inform travel restrictions or other interventions. Genomics provides a detailed picture of disease dynamics that traditional methods can't match. Genomics isn't just about the pathogens, it's also about the patients. By understanding genetic differences in individuals, doctors can tailor treatments to be more effective. This personalized approach is becoming increasingly important in infectious disease management, especially for complex or resistant infections. International collaboration is essential for tracking infectious diseases. Genomic data is shared across borders, allowing scientists to monitor global trends and respond to threats more quickly. These networks have become a backbone of modern public health, enabling coordinated action against emerging diseases. The vast amount of data generated by genomics requires powerful computational tools. Bioinformatics is the field that turns raw genetic sequences into actionable insights. By using algorithms and databases, scientists can identify patterns, predict outcomes, and guide public health decisions. With great power comes great responsibility. Genomic data can reveal sensitive information about individuals and communities. Ethical guidelines are essential to protect privacy, ensure informed consent, and prevent misuse of data. Balancing scientific progress with ethical considerations is a key challenge in this field. Advances in technology have made sequencing devices smaller and more portable. Scientists can now bring these tools into the field, enabling rapid analysis during outbreaks, even in remote locations. This mobility is transforming how we respond to infectious disease threats. Genomics isn't limited to clinical samples. Scientists also analyze environmental samples like water, air, or surfaces to detect pathogens. This approach helps identify sources of outbreaks and monitor the effectiveness of interventions in real time. Many infectious diseases originate in animals before jumping to humans. Genomics helps trace these zoonotic events, revealing how pathogens cross species barriers. This knowledge is crucial for preventing future pandemics and understanding the complex relationships between humans, animals, and the environment. Metagenomics is the study of genetic material recovered directly from environmental samples. Instead of focusing on one pathogen, scientists can analyze entire communities of microbes. This approach uncovers hidden threats and provides a broader view of infectious disease dynamics. Genomic technologies have made it possible to diagnose infections faster than ever before. By detecting pathogen DNA or RNA directly from patient samples, doctors can start the right treatment sooner. Rapid diagnostics are especially important in controlling outbreaks and improving patient outcomes. Genomic epidemiology combines genetic data with traditional epidemiology to track disease patterns. By linking genetic sequences to patient information, scientists can uncover transmission chains and identify risk factors. This integrated approach is reshaping how we study and control infectious diseases. Open access to genomic data accelerates research and improves public health responses. Scientists around the world share sequences and findings in real time, fostering collaboration and innovation. This spirit of openness has been vital in recent outbreaks, enabling faster solutions. Not all regions have equal access to genomic technologies. Low-resource settings may lack equipment, trained personnel, or reliable internet. Addressing these gaps is essential for global health equity and effective disease control everywhere. The field of genomics is constantly evolving. New technologies like single-cell sequencing and CRISPR are expanding our capabilities. These innovations promise even faster, more precise, and more affordable tools for infectious disease research and control. Genomic data informs public health policy at every level. From local outbreaks to global pandemics, decision-makers rely on this information to guide interventions, allocate resources, and communicate risks. Effective policy depends on accurate, timely genomic insights. As genomics becomes more central to infectious disease control, education and training are crucial. Scientists, healthcare workers, and policymakers need to understand genomic data and its implications. Ongoing training ensures that new technologies are used effectively and responsibly. The One Health approach recognizes that human, animal, and environmental health are interconnected. Genomics supports this holistic view by tracking pathogens across species and ecosystems. Collaboration across disciplines is essential for preventing and controlling infectious diseases. With advances in sequencing and data sharing, real-time surveillance of infectious diseases is now possible. Scientists can detect new threats as they emerge and respond quickly. This agility is key to preventing outbreaks from becoming epidemics or pandemics. During the Ebola outbreaks in West Africa, genomics played a critical role. Sequencing the virus helped trace transmission chains, identify sources, and guide control measures. This experience demonstrated the value of genomics in managing deadly diseases. Genomics enables the discovery of new pathogens that might otherwise go undetected. By sequencing unknown samples, scientists can identify novel viruses or bacteria, expanding our understanding of the microbial world and preparing us for future threats. Protecting genomic data is essential. Security measures must be in place to prevent unauthorized access or misuse. As more data is generated and shared, robust systems are needed to safeguard sensitive information and maintain public trust. Citizen science projects invite the public to participate in genomic research. By collecting samples or analyzing data, non-scientists can contribute to infectious disease surveillance and discovery. This engagement broadens the impact of genomics and fosters community involvement. Travel can accelerate the spread of infectious diseases. Genomic surveillance at borders and airports helps detect and contain threats before they spread widely. This proactive approach is vital in our interconnected world. Accurate genomic analysis starts with proper sample collection. Whether from patients, animals, or the environment, high-quality samples are essential for reliable results. Standardized protocols ensure consistency and accuracy in genomic studies. Interpreting genomic data requires expertise. Scientists must distinguish between harmless variations and significant mutations. This careful analysis guides public health actions and ensures that interventions are based on solid evidence. Infectious disease genomics brings together experts from many fields: biology, medicine, computer science, and public health. Collaboration is essential for translating genomic discoveries into real-world solutions. Working together, these teams can tackle complex challenges more effectively. The future of infectious disease genomics is bright. Advances in technology, data analysis, and international cooperation will continue to enhance our ability to detect, track, and control diseases. Staying ahead of emerging threats requires ongoing innovation and investment. Social media platforms can amplify the impact of genomic discoveries. Sharing information about outbreaks, new variants, or research findings helps keep the public informed and engaged. Responsible communication is key to building trust and encouraging positive action. Despite its power, genomics has limitations. Not all mutations are well understood and data interpretation can be challenging. Uncertainties remain, especially with new or poorly studied pathogens. Ongoing research is needed to fill these gaps. While costs are decreasing, genomic technologies can still be expensive and complex. Making these tools accessible to all regions and communities is a priority for global health. Efforts are underway to reduce costs and simplify processes. Genomic research is subject to legal and regulatory oversight. Laws govern data sharing, privacy, and the use of genetic information. Navigating these requirements is essential for responsible research and international collaboration. Engaging communities in genomic research builds trust and improves outcomes. Involving local leaders, respecting cultural values, and communicating clearly are all important. Community engagement ensures that research benefits everyone. Biobanks store biological samples for future research. Genomic analysis of these samples can reveal long-term trends and support retrospective studies. Biobanks are valuable resources for understanding infectious diseases over time. Artificial intelligence (AI) is increasingly used to analyze genomic data. AI can spot patterns, predict outbreaks, and suggest interventions faster than traditional methods. This technology is making genomics more powerful and accessible. From tracking Ebola to controlling COVID-19, genomics has delivered remarkable successes. These stories demonstrate the real-world impact of this science, saving lives and shaping the future of public health. Continued investment will bring even more breakthroughs. Infectious disease genomics is a rapidly advancing field with enormous potential. As technology improves and our understanding deepens, genomics will play an even greater role in protecting global health. The journey is just beginning, and the future holds exciting possibilities for science and society.